Welcome back, focused learners, to today's explainer. I am absolutely thrilled for this one because we're about to go on a journey of seriously epic proportions. We are going to travel all the way from the massive macro scale of life's entire history on Earth, zooming straight down into a microscopic, incredibly fragile barrier that is literally dictating your survival right at this very second. But before we get out our microscopes, I want you to consider a pretty high-stakes question. What do the grand evolution of life on Earth and a deadly chemical weapon like sarin gas actually have in common? I know, it sounds like the setup to a really strange riddle, right? Okay, let's dive into this. The answer actually lies in cellular biology. It seamlessly connects the absolute largest evolutionary leaps in history directly to the tiny molecular mechanisms of life and death. So here is our roadmap for our macro to micro journey today. First up, the two classes of cells. Second, the cell's border control. Third, the anatomy of a membrane. And fourth, we'll zoom all the way down into the lipid building blocks. Let's kick things off with section 1, two classes of cells. This right here is the fundamental evolutionary division in all of biology. Now, to understand the sheer scale of this division, you kind of have to look at Earth's biogeologic clock. The Earth itself originated around 5 billion years ago. If we jump forward, early, simpler eukaryotic organisms showed up well over 1.5 billion years ago. But notice how recently complex multicellular animals actually appeared in the fossil record, only about 600 million years ago. In the grand scheme of things, that is a really sudden arrival. In us humans, we just barely showed up at the very, very end of this timeline. And this brilliantly illustrates the massive gap between these two groups. What separates ancient, simpler life forms from the complex life we see today? Well, you have prokaryotic cells, which are smaller, structurally simpler, and contain just a single circular chromosome tucked into a kind of poorly demarcated area called a nucleoid. In fact, prokaryotic literally translates to before the nucleus. Then you have eukaryotic cells, meaning true nucleus. These guys are much larger, incredibly complex, and house separate, linear chromosomes securely enclosed inside a complex membranous nuclear envelope. And because eukaryotic cells carry a considerably larger amount of genetic information, their DNA needs some seriously specialized packaging. I mean, a simple bacteria might have a few million base pairs of DNA, but eukaryotics have vastly more. To handle all that data, eukaryotic chromosomal DNA is tightly bound up with proteins to form a complex nucleoprotein material known as chromatin. Think of it like a brilliant, highly organized biological filing system. Moving right along to section 2, the cell's border control. Let's talk about the dynamic plasma membrane. Now, right off the bat, I want you to completely toss away the misconception that a cell is just surrounded by some static hard wall. The reality is so much more dynamic than that. The plasma membrane, that vital outer boundary separating the living cell from the non-living world, is only 5 to 10 nanometers wide. Think about that. That's thousands of times thinner than a single strand of spider silk. You literally can't even see it with a traditional light microscope. Yet, this unbelievably tiny, fragile structure is all that stands between a cell's survival and its total destruction. And this membrane is a multitasking marvel. It is an active border control and a communication hub, not just a passive barrier. It handles compartmentalization, enclosing distinct intracellular spaces, so specialized activities can happen without interfering with each other. It actively transports solutes, pumping vital sugars and amino acids, often fighting against the current from low to high concentration. Through signal transduction, it acts like a cellular antenna, responding to external stimuli using highly specific receptors. It even handles energy transduction, like converting sunlight into chemical energy in plants. And it's doing all of this while being less than 10 nanometers thick. Unbelievable. Which brings us to section 3, anatomy of a membrane, how lipids and proteins exist in perfect harmony. So exactly what kind of materials make up a 5 nanometer border control capable of such mind-blowing tasks? Well, when scientists first got to look through early electron microscopes, they stained cell tissue with heavy metals like uranium and lead. These metals naturally bound to the polar surfaces of the membrane, and it revealed this very distinct 3-layered or trilaminar pattern, 2 dark lines on the outside and a lighter space right in the middle. This gave us our first solid visual proof that the membrane is essentially a bimolecular layer, a double sheet. So the crucial point is really perfectly captured in this quote from our sources. The lipid bilayer serves primarily as a structural backbone. The proteins of the membrane, on the other hand, carry out most of the specific functions. Basically, the lipids build the physical barrier, stopping water-soluble materials from just leaking in or out, while the proteins act as the specialized workers, managing all those complex operations we were just marveling at. But keep in mind, this lipid-to-protein ratio isn't just some standard one-size-fits-all formula. In biology, form follows function entirely. Take the inner mitochondrial membrane, for example. It handles cellular respiration and is absolutely packed with proteins for the electron transport chain, meaning its ratio of proteins to lipids is very high. On the flip side, consider the myelin sheath membrane that wraps around your nerve cells. Its primary job is electrical insulation, so it needs a really thick lipid layer with very minimal protein. The membrane completely customizes its makeup based on exactly what specialized activity the cell needs to do. And that brings us to our final, most magnified stop on our macro to micro journey. Section 4, the lipid building blocks, the literal chemistry of survival. The foundational secret to how these membranes can form stable sheets in a watering world comes down to one amazing word: amphipathic. Every single membrane lipid possesses this sort of dual personality. They all have a hydrophilic, or water-loving region, alongside a hydrophobic, or water-fearing region. Because our cells are swimming in a watery environment, the water-fearing tails naturally hide in the middle, while the water-loving heads face outward. Boom! They automatically assemble into those stable, double-layered sheets. Now, we generally break these amphipathic membrane lipids down into 3 main types. First up, phosphoglycerides, which are by far the most common type, built on a glycerol backbone. Next, we have sphingolipids, which use a sphingosine backbone and are super important in your nervous system. And finally, cholesterol, found specifically in animal cells, which is made of sterile rings that give it a unique, rigid structure. Let's zoom in on that most common group, phosphoglycerides. They rock a 3-carbon glycerol backbone along with 2 fatty acyl chains. You've probably heard tons about the health benefits of EPA and DHA, the highly unsaturated omega-3 fatty acids found in fish oil. Well, here is the real world so what. These specific lipids are quite literally woven directly into the membranes of your cells in high concentrations, particularly in your brain and your retinas. What you eat literally becomes the border control of your most vital organs. Let's move to and see how this builds for our second category, sphingolipids. These are basically assembled like biological Legos. You start out with an amino alcohol called sphingosine. Then you snap on a fatty acid, which creates a ceramide. Finally, if you stick a carbohydrate on top of that, you get a glycolipid, like a ganglioside. As I mentioned, these modular lipids are especially crucial for your nervous system's proper function. But there is a much darker medical reality to these glycolipids. Since they sit right on the surface acting as precise chemical receptors, sometimes the wrong things recognize them. The deadly toxins that cause cholera and botulism, and even the influenza virus, actually invade target cells by binding specifically to these cell surface gangliosides. These microscopic lipid structures are the exact, specific entryways exploited by these lethal diseases. And our final key membrane lipid is cholesterol. In certain animal cells, a staggering 50% of the lipid molecules in the plasma membrane can be cholesterol. Because these molecules are flat and rigid, they pack themselves right in among the squiggly fatty acid tails, which slows down their movement. This effectively stiffens the whole bilayer while somehow, magically, still maintaining its overall fluidity. It's a brilliant balancing act. And that brings us perfectly full circle to the profound thought we started this explainer with. Think back to that sarin gas. Sarin operates by poisoning a single specific enzyme that normally breaks down chemical signals sent to your muscle cells. Without that enzyme functioning at the membrane, the muscles you use for breathing become constantly stimulated until they are completely paralyzed. It proves that the most massive stakes imaginable, life, death, and complex evolution itself, all rely entirely on a fragile microscopic border just 5 nanometers wide, which really leaves you wondering, what other hidden vulnerabilities are sitting right there on the surface of our very own cells?